There are 4 bases, but 20 amino acids, including glycine, tryptophan and arginine. Humans can synthesize 10 of these, but we must consume the others in our diets. Phenylalanine is one such example of an essential amino acid. The amino acids encoded link together via peptide bonds to form polypeptide chains. This sequence of amino acids is known as a protein's primary structure. Just as long DNA molecules can form helices, amino acids can also form such structures in a protein's secondary structure. Secondary structures include alpha helices and beta sheets. A combination of these domains form tertiary structures such as jelly rolls and beta barrels. Combining these subunits results in the final quaternary structure of a complete protein. The chain of amino acids is held together by strong covalent bonds. Secondary, tertiary and quaternary structures are held together by weaker intermolecular forces such as van der Waals forces, electrostatic attraction and hydrogen bonds. A protein structure is integral to its function. Clefts in a protein surface can act as important binding sites for other proteins. Binding sites on an antibody, for instance, allow it to bind to a virus and target it for destruction by other immune cells. Other structures may act as triggers for signalling processes. As a protein is bound by a signalling molecule, it may change shape, which in turn activates another process downstream. Events like these are involved in our senses of taste and vision. X-ray crystallography is a powerful modern technique that has allowed scientists to examine these 3-dimensional structures. This method involves firing a beam of X-rays at a protein crystal. As X-rays hit electrons in the atoms, they are diffracted, and this diffraction pattern can be analysed to determine the structure of the protein. Determining protein structure has elucidated many biological processes and is helping to refine our understanding of human health and disease as a result. So now we know what our DNA is and what it does, but where is it found in our bodies? It's actually found in the nucleus of every single cell in the body, apart from the red blood cells. Fitting all this genetic information inside the nucleus of a single cell is very difficult. In fact, it is the equivalent of trying to fit 24 miles of thread into a tennis ball. Therefore, a complex system is required to efficiently package DNA into each cell. Chromosomes offer a method in which genetic information can be condensed and compacted effectively. They often exist in pairs. Each half of a chromosome is called a chromatid, and 2 sister chromatids are joined together at the centromere. Whilst different organisms have different numbers of chromosomes, human DNA is packed into 23 chromosomal pairs. This includes 22 autosomal pairs, with 11 chromatids, each from the mother and father, and 2 sex chromosomes, XX for females or XY for males. The complete set of chromosomes for an organism is known as its karyotype. A chromosome consists of 3 important features: a replication origin, centromere and telomere. The replication origin is the site where DNA replication begins and is of major importance during cell division. The centromere joins 2 sister chromatids together and is therefore important following cell division when the chromatids are separated. Finally, the telomere consists of a highly repeated genetic sequence at each end of a chromatid. This allows it to be efficiently replicated to a daughter cell and protects it from being damaged. Shortening of the telomeres has been thought to be the cause of aging. In recent scientific studies, the enzyme telomerase has been used to lengthen telomeres in mouse DNA. This halted the aging process and in some cases reversed it. However, due to an increase in the production of cancerous cells as a result, it will be some time before this process is mastered by humans. Each chromosome consists of tightly wrapped DNA. Just like tightly wrapped string can form a ball, but it is more complex than this. Chromosomal structure is arranged in levels. Firstly, nucleosomes are formed by complexing DNA with proteins called histones, along with RNA molecules needed for gene expression, DNA replication and repair. The DNA is wrapped around these proteins like wrapping a section of string around the ball. Free DNA links nucleosomes together to form chromatin, which is commonly referred to as beads on a string, with sections of DNA and protein as the beads, and free DNA as the string. Nucleosomes served to reduce the original length of a DNA molecule. Secondly, nucleosomes are wrapped together into a chromatin fibre. Finally, this chromatin fibre is wrapped up again to form the final chromosome. This complex packaging system allows a vast amount of genetic information to be stored in our cells, but it is also highly important that this information is easily accessible to allow DNA to be manipulated in order for gene expression, DNA replication and repair to occur. Eukaryotes, like humans, possess a variety of ATP-dependent chromatin remodeling complexes, which can make DNA less tightly bound to the histone protein. This makes the DNA more accessible to modification by other proteins. Heterochromatin is a highly compacted form of chromatin, and any genes in these regions are silenced and not expressed. Euchromatin, on the other hand, comprises of less compacted chromatin, in which genes are readily expressed. Just think of it as though you want to access certain parts of a DNA molecule, as you may wish to read a page of a book. If the page was in a book that was neatly packed away onto a busy bookshelf, it would be difficult to get access to the page you wanted to read. But if the book was taken off the shelf, it would be much easier to access the book and read that page. DNA must be modified in order to be repaired or for genes to be expressed. If it is tightly wrapped up by other proteins, it cannot be accessed, so loosening it up enables this modification to occur. Silencing a gene enables DNA to be neatly packaged whilst the gene is not needed, whilst also protecting it from damage or unnecessary modification. Now we know how our DNA is packed into our cells, let's learn more about our genes.